Hi, I'm Christy Avery. I'm an associate professor at the University of North Carolina. And today it's my distinct pleasure to introduce Dr. Brant Levitt, the Genomic Data Science and GWAS Data Coordinator for AdHealth. Brant joined our AdHealth team 3 years ago, and as you'll see, he's built an impressive pipeline for the analysis and interpretation of AdHealth omics data. Please remain muted and enter questions into the chat for me to read out loud after Brant's presentation. Take it away, Brant. Thank you so much for doing this.
No problem. Happy to do it. All right, let me see if I can share my. All right. Thank you guys all for taking the time to be here and allow me to talk about some of my work with the AdHealth genetic data and the Genome-Wide association study data set in particular. About a year and a half ago in Nature, there was this opinion piece on the heels of a paper that looked at the social stratification of the former coal mining industries of north central England. They noted that in this area, Yorkshire in particular, the last of the coal mines had closed down, and there were these populations of people who the modern economy had really just left behind in the dust. And in this paper, they did a GWAS to try to understand from their perspective what sort of biological implications there might have been in their inability to adapt to the modern economy. They applied some of the findings from a GWAS, Genome-Wide Association study, that looked for gene variants associated with fewer years of educational attainment, and found that these variants were highly clustered in these populations of former coal miners, and might have gone on to explain a little bit of the blue-collar nature of this area. And so in my mind, this seems like a good motivating example of how social scientists can make use of genetics and genetic data to deepen their understanding of their subject matter. In the article, they had a quote that I thought was particularly useful for this presentation, and it goes to the geneticists and social scientists doing the work. The results offer a useful and important guide to the relative contributions of nature and nurture to specific behavioral traits. Just as genetic analysis can already highlight people who have an increased risk of cancer or heart disease, the approach could, for example, improve understanding of how the environment affects complex traits, and so offer a way to intervene to improve areas such as public education. And in the intervening years, since 2019, when that was published, there have been several other high-profile GWAS studies focusing on social traits that have come to the fore, including, well, the educational attainment study, one on same-sex attraction, another on loneliness, one on aggression and antisocial behavior, another consortium on smoking, and another on mood disorders, particularly bipolar disorder. And so with that kind of background, I'd like to jump into my presentation, where I'm going to describe in a little greater detail what a GWAS actually is and why a social scientist might want to conduct one, what sort of data goes into it and what you do with the data, and what the outcomes from that data actually look like. I'll address where these sort of results end up and how they can be used, and then kind of give you a motivating start of where you might go to learn more and eventually use this in your own research. Parenthetically, this presentation was written presuming that you're a social scientist and you're interested in incorporating genetic data into your research in some way. So let's start with a little bit of background about what a GWAS is. Simply, a GWAS is looking for genetic variants that are overrepresented in people with your outcome of interest. In this cartoon, we have little orange people defined as cases with our outcome of interest, or controls without our outcome of interest. And below them, we have cartoon schematics of chromosomes. And in red, we have a gene variant. And if we just count up the number of chromosomes in the people with our outcome of interest, we see that the frequency of this red gene variant on chromosomes in our cases is at a much higher rate than in the general population. And so while there's a lot more technical information that goes into it than that, this forms the basis for a GWAS study. And so let's talk a little bit about what a genetic variant is. Humans are comprised of cells which, with very few exceptions, contain a nucleus containing sets of chromosomes, which are aggregated bits of DNA wrapped around structural proteins. The chromosomes are shaped roughly like this and are composed of DNA. I'm showing you a karyotype here. This is microscopic images of chromosomes. Each human has 23 pairs of chromosomes. 22 of them are autosomal and 1 pair is a sex chromosome. The DNA itself is made up of a sugar ribose backbone that's held together by rungs of adenine, guanine, cytosine, or thymine, which we shorten to A's, T's, C's, and G's. And bioinformatically, we just list them as in this example on chromosome 19 at site 44,900,000 whatever. In the bulk of the population, this region goes C, G, A, T, A, T, T, C, C, and then there's a base that varies in some of these people in our imaginary little study right here. In the first individual in our imaginary study, on one of their chromosomes, they have the normal C, but on the other chromosome, they have a variant, a gene variant, a T. The second person has the normal C on both chromosomes. Third person has the gene variant T on both chromosomes, and so on. And so we would say that these people at this SNP have a genotype. Individual 1 is a genotype of TC, individual 2 has CC, individual 3 has TT. As it turns out, chromosome 19 at base 44 million whatever isn't just some random site. It's a site that is part of a gene called apolipoprotein that is highly linked to Alzheimer's, and having 2 of the risk variant alleles, the T's, puts you at a higher risk of developing Alzheimer's compared to if you only have one of a T on one of your chromosomes or no T's at all. And so while I've just described a substitution type gene variant, there are others that we keep track of, insertions and deletions where a base may be added or missing. We term those indels. Whole stretches of DNA may be added or deleted or switched around. There also may be gene duplications or inversions. And the sites at which these gene variants occur also play a role in whether or not they hold biological and thus social function for the individual. This cartoon is just a schematic of the structure of a gene. It includes a way upstream of the gene. There are regulatory regions, enhancers and silencers that other things bind to promote the expression of the gene or stop the expression of the gene. There are other regulatory pieces that are involved in the mechanics of gene expression. And the gene itself is divided into several regions that come together in different ways to produce slightly different proteins. And so gene variants may occur in the coding region in these red parts and disrupt the function of a protein directly, or they may fall in a splicing area and limit the construction of a certain form of the protein, or they may stop a protein from being expressed in the first place. So these gene variants can have all kinds of different effects depending upon where they are. And the effect of those gene variants can be visualized on this graph on the y-axis, with low effect on bottom, high effect on the top, and the prevalence of those gene variants in the population is on the x-axis, going from very rare to very common. Now, a rare gene variant that causes a huge effect is oftentimes a Mendelian disease. This is often what we think of when we think of genetic diseases, metabolic disorders, where if you have the gene, you have the disease. But there are also a lot of variants where it takes the additive effect of many common variants in order to add up to causing the disease. And this is really where GWAS shine. And I'm going to park this presentation here for just a moment so I can address something. There are going to be rare variants that have a lower impact on disease. Now, these are going to be harder to find in a small population, which is why oftentimes GWAS data sets will include hundreds of thousands of people, so that you can find either very rare variants or variants that have very small effects on their own. That doesn't mean, though, that a smaller sized GWAS data set is useless. It just means that it's best for finding variants of middling effect and require looking at the right populations with the right questions. So now that we know a little bit about what a GWAS is, why don't we talk about why we conduct one? Well, the typical reasons can be divided roughly into the academic reasons, understanding the biological underpinnings of an outcome of interest, or reconstructing the species evolutionary past, or doing a family ancestry and understanding more about an individual. Alternatively, there are biomedical reasons why people do GWASs, to identify drug targets, to identify biomarkers for prognostic or diagnostic uses, to subtype diseases and understand likely symptoms. And finally, there are public health reasons, identifying at-risk populations, hoping to prescribe preventative practices to people before they go on to develop a negative outcome. GWAS would also be useful for genetic counseling and newborn screening or prenatal screening. But for a social scientist like yourself, what can GWASs do for you? I've thought of a few examples. One is you can identify what proportion of your outcome is actually due to the subject's genes, and then inversely, how much is not. You could identify certain populations of interest that are going to be more or less likely to develop an outcome. And we'll talk about polygenic scores a little later and how those can be useful for that. We might also want to understand if there are biological factors that mediate or mitigate this relationship that you're studying between an exposure and an outcome. You might also want to look if there are shared genetic predispositions between your outcome and other outcomes to make cognitive links between different areas of study. And finally, you might want to look to see how your subject's environment goes on to impact your outcome of interest, and if the environment conspires in some way with the host genetics that you might not necessarily predict. And so I've told you a little bit about the rationale for why you might want to do a GWAS. So I thought at this point it might be fun to see the frequency with which people are actually doing them in recent years. This is from the GWAS catalog. It's a little video that shows the 23 chromosomes and shows over time GWASs that have been done, and it'll have little circles that identify places in the genome on these different chromosomes. that are significantly linked to whatever outcomes being studied. And so we see starting in 2007, GWAS has started to become more commonplace. And over the last decade, there's been a real explosion in the number of traits that have been linked to different genetic areas. And I think this taps out at around 2019 or so, but it definitely, studies definitely did not stop being done at that point. So I'll show you a few examples of GWASs that were done using social outcomes that might be of interest. And these were all done in the last couple of years. And that's also meant to reinforce that this is still an area of active investigation. One of my favorite papers to come out the last couple of years was this one by folks using the UK Biobank. They wanted to understand social isolation. And so they asked people about their feelings of loneliness and their involvement in religious and sports groups and how frequently they go out to pubs. You know, they're English, so going to pubs is an important part of their social life. And they look to see if there are gene variants then associated with each of those 4 domains of this signature of isolation. And they plotted on what's called Manhattan plot. These Manhattan plots kind of look like the skyline of Manhattan with you know, tall buildings. On the x-axis are the 22 autosomal chromosomes, and on the y-axis is the negative logarithm of the p-value. And each dot here represents a different gene variant. And so if a gene variant is highly linked to the outcome of interest, you'll see its negative log, its p-value goes higher. And so we find that they find that there were several gene variants and areas of the genome linked to loneliness. Similarly, there were several areas linked to lack of involvement in a religious group and sports group. And so from this, they were able to look at overlapping gene variants and find genes associated with social isolation. Another recent example that I think does a good job of showing the promise of GWASs for social outcomes was this one. This is a consortium to which actually AdHealth contributed data that looks for antisocial behavior, and specifically looks for gene variants associated with antisocial behavior that are different between men and women. Because as it turns out, they find that men and women have entirely different pathways of antisocial behavior. Another recent study was the educational attainment study I had mentioned earlier that was in part authored by former AdHealth genetics personnel that found several different sites linked to number of years of educational attainment. And finally, some of the same individuals involved in that one also recently published a GWAS study for same-sex attraction that found that there really aren't any clear genetic links between same-sex attraction. There aren't very many clear genetic links to same-sex attraction, so that it's something that is more heavily influenced by your environment. So hopefully I've motivated you a little bit about why social scientists might want to conduct a GWAS and shown you some examples of how it's worked out successfully. But let's delve into the nitty-gritty and look at the kinds of data that gets used. The data that AdHealth collected was done by consenting participants, collecting saliva from them, extracting DNA, and applying that DNA to an omni-genotyping chip. Now, this chip gives us information at, well, this one in particular gives us information at 1 million different gene variants. You know, if they have which alleles the person has at each of those variants, and that's visualized using fluorescent cameras. The data itself ends up in what's called a Plink format. These are human readable files that are pretty easy to parse and have a lot of tools that can be used with them. We'll focus first on these upper to the PED and MAP files. The PED files have a family ID, an individual ID, father's ID, mother's ID, sex, outcome, and then a different column for every single gene variant that's being studied. And within, and each row is a different person. And so if we look in the PED file at this very first gene variant, this RS1, we see for this first person it's CT, for the next person it's CC and CC, but it's different for each gene variant. And then the map file gives us basically a rundown of the gene variants that were shown, what chromosome they're on, what they're called, their base position in the genome, and something that gets rarely used, a genetic distance. So these are converted into a non-human readable format consisting of FAM, BIM, and BED files that break apart the stuff about the participant itself, stuff about the gene variant itself, and the genotyping in the BED file. And then you can also encode covariates that you might find useful for your study. So once we have this data in Plink format, we did some quality control first on the participants by removing individuals with low call rate or poor concordance with their duplicates, or sex chromosome discordance, or identifier abnormalities, and got almost 10,000 participants that passed quality control. Additionally, there's also some quality control on the variants. There were two different chips that were used, one with million variants, one with 2.5 million variants, and some variants were removed for failure to map to the genome. Some were removed for having multiple different alternate alleles. Some had low call rates. Some were removed for being particularly rare, and some were removed for being under active evolution. Some were removed for having poor duplicate discordance. And then variants common to both chips were collected, leaving us with about 600,000 gene variants. Apologize, the cat found his way in. So once we had these 10,000 people with 600,000 gene variants, we did an imputation, genomic imputation process that goes from genotyped gene variants across the genome, shown in like this blue chromosome right here, where we don't necessarily know the status of the gene variants that weren't measured that are between the ones that were. But by using empirically found haplotypes, that is stretches of commonly inherited gene variants together, we were able to make a prediction about what the gene variants at the different sites between measured variants were going to be. So instead of just having A, G, and C in this blue chromosome, we were able to align it to reference haplotypes and figure out that it goes A, TT, G, TT, C. And we have an imputation quality score for each of the gene variants that we imputed. So that imputed data ends up in a file format that's a human readable format called variant call format, VCF, that contains headers with lots of metadata about each variable, including chromosome position, name, reference, and alternate alleles, some quality information, and then the genotypes with each sample having a different column. And so, for instance, for this first gene variant, for the first sample, we have no minor alleles on one chromosome and no minor alleles on the other. So that would be GG. But for the same gene variant for sample number 2, we have a risk variant, a minor allele, and a non-minor allele. So we'd have GA as the genotype for this person, and so on. And so if we look at the kinds of people that make up this 10,000, this is an area where AdHealth really has a lot of strength. One of the big criticisms of the world of genomics and GWASs in general is that they've had a myopic focus on samples of convenience, that is largely white European people. However, the Ad Health sample set includes a good number of Asian and Pacific Islanders, non-Hispanic Blacks, Hispanics, Native Americans, and so on. And there's roughly an equal distribution of males and females, and the ages at which samples were taken kind of cluster around early middle age. In addition, and you might have heard about this in the plenary sessions, one of the things that's really cool about the Ad Health data set is because of the sampling structure, we have nested genetic studies. We have monozygotic twins who share 100% of their genome. We also have dizygotic twins and full siblings and half siblings and cryptically related people. And this breeds the ability to do all kinds of twin studies and adds to the complexity of what we're doing in some neat ways. So now that we know what kind of data we're talking about, how do we analyze it? Well, we talked about the importance of the non-random association of gene variants when we were doing imputation, and those are frequently very much tied to ancestry groups. So we can reduce the complexity of this genetic data using a multidimensional scaling approach called principal component analysis. And when we look at just the first two principal components, 1 and 2 plotted against one another with each dot being a different person, we see that there's a pretty substantial difference in by genetic ancestry. We see that non-Hispanic white people in purple seem to cluster up here, and I have them picked out in this graph. When we look at non-Hispanic Black people, they tend to cluster up here, which I've picked out right here. For Asians, they cluster down here, and Hispanics seem to cluster in green over here. And we can use those principal components to account for population structure. Additionally, in order to do the analysis, we need to decide upon our outcome of interest. And that can be binary dichotomous yes-no kind of variables, or it could be a measured quantitative variable, or it could be ordinal, hot, hotter, hottest kind of measure. Could be categorical, where there's no internal order to the categories. And another one of the values of this Ad Health data set is it's longitudinal. So we can have outcomes that are trajectories over time, or we can look at time to the occurrence of an event as an outcome. Whatever the case is, we'll generate a phenotype file, which I'll show you next, that feeds into an association algorithm that we like to use called SUGEN. This is great because this allows for a family level correction, which accounts for those nested genetic study structures that I described, and also accommodates the racial variation of the sample that I described by giving you the option of a heteroscedastic variance flag. It uses those VCF files that I described as the input and allows you to do either GWAS or a gene by environment study. So first thing we need to do in order to use SUGEN with our VCF files to do our analysis is create a phenotype file. Now this is going to have a family and individual ID. It's going to have all the demographic covariates that you think are going to be important to your question. Here I just have sex, age, and race listed. It's going to have statistical weights and principal components, and then your outcome of interest. So in order to run the GWAS in a command line environment, we're just going to tell the computer where we've installed SUGEN, where we've saved that phenotype file, the IDs for the family and the individual ID, where we've saved the VCF files, the formula that we want, so the outcome, the covariates and the principal components, and then give it that heterovariance flag, and then tell it where we want it to save its output. And so what does that output actually look like? Well, it looks like this human readable file that contains variables with one row per gene variant, with variables for chromosome, position, the name of the variant, reference and alternate alleles, how frequent the alternate allele is in your population, population size, the effect size, the beta, standard error associated with beta, and the p-value, how tightly associated that gene variant is with your outcome of interest. And so then we'd go on to filter those gene variants, getting rid of ones that have that are very rare in our population, or have questionable imputation, or the population size presents some statistical vagaries. But the upshot is we still have millions of statistical tests, and we need some way in which to correct for that multiple testing. And so a rather conservative way of doing that is the Bonferroni correction, which takes your significance level and divides it by your number of tests. So for 1 million tests, for example, you'd get a corrected p-value of 5 times 10 to the minus 8. So that usually tends to be the genome-wide significance level. But as I said, it's a bit conservative, and so a lot of the software will look for things between 10 to the minus 5 and above as being suggestive. So once we have that human readable file that I just shown you, we can use an R package called QQman to visualize some of the results. Here we're visualizing a quantile plot. This demonstrates the level of overinflation or underinflation that we have in our association model. On the x-axis, we have the expected minus logarithm of the p-value, and on the y-axis, we have what it actually is. So most of our variants, of course, are going to be down here. We aren't going to expect that they'd be significantly associated, and they're not. But as we move up this x equals y line, there are going to be some that wildly exceed what we expected. These ones are significantly linked to our outcome of interest. And we can reduce then the QQ plots down to a metric called a genomic inflation, a lambda value, and be able to compare GWASs between one another for over and under inflation. Additionally, we can use that same QQman library and just call the function Manhattan and generate the Manhattan plots that we were already looking at. And so in this example, it has alternating black and gray for each of the chromosomes on the x-axis, a different dot for each of the variants, a blue line for that suggestive 10 to the minus 5 p-value, and a red line for that 5 times 10 to the minus 8 genome-wide significance value. And we see that there's a clear peak in this example in chromosome 3. This is clearly linked to our outcome of interest. And finally, we might want to visualize the hits in greater detail using another command line program called Locus Zoom that takes in that same data and plots each of the circles as each of the gene variants as a circle, except the lead variant that's most highly associated, which is a diamond, and the association based on linkage disequilibrium between the lead SNP and the other ones around it are shown in a gradation of red down to dark blue. And this is all superimposed on the chromosomal coordinates where we can see individual genes in the genome. And so in this example on chromosome 19, we see that there are several gene variants that are highly associated with the outcome in this case being smoking at a gene called CYP2A6. It's a cytochrome gene. And so the authors that had done this one concluded that this gene is linked to smoking behavior. So when we consider these sorts of outcomes that we get from these GWASs, we might consider the number of significant hits that we get, or we might see if those hits are clustering within one gene or a small number of genes. We may also look at the degree of significance. Are they at 10 to the minus 8? Are they at 10 to the minus 20? That makes a pretty big difference. We might also look at the strength of association, the effect size. We might also look to see if there are several related genes that were all hit in our GWAS. And we may also look to see if there's a clear functional effect of some of our gene variants. And so as we interpret these kind of results, one of the things we can do is we can look for a shared genetic etiology between our outcome of interest and other people's GWASs. So for example, in this paper, they did a GWAS of neuroticism and found the linked gene variants overlap pretty highly with those involved in anxiety disorders and depression and ADHD and anorexia, but less so with age of having your first child or IQ or educational attainment. Additionally, because each of those genes are annotated, we can look to see if there's an enrichment of hits in specific biological processes and be able to link a biological process or pathway to an outcome of interest. Finally, another way in which people might interpret GWASs is they'll look at what are called EQTLs. These are genes whose expression depends upon whether or not that gene variant is present. And they'll look for the gene variants that they find, they'll look to see if the expression of those genes is more or less common in certain anatomical regions. And so in this study, they did a GWAS of household income, found several different genes that were highly linked, and then found that those genes were expressed very highly in certain areas of the developing brain. All right, so now that we have a feel for what sort of outcomes we can get, where do they go? Are there repositories for them? And yeah, obviously I wouldn't have said that if there weren't. First I'll tell you about is the GWAS Central. And that looks like this. And we can type in our traits of interest. This case, I just typed in loneliness and look at what sort of studies have been involved, and within those studies, where the associated gene variants lie on the chromosomes. Additionally, we might want to look at the GWAS catalog, which is a European repository, and I'm just searching mathematical ability because I think that's kind of fun. And we can pull up, when this loads, we can pull up individual associations and look for genes that are tied to mathematical ability. But you may end up having to do that on your own. And then the last one I'd like to draw your attention to is, oh, here it is. This was the GWAS catalog. Shows the name of the SNPs, how highly linked they are, betas, confidence intervals, mapped genes, and all of this is available programmatically as well. So you, instead of trying to copy down 2,700 associations, you can download what you need. So FENGENY is the last one I'd like to draw your attention to, and I'll look for the trait social responsibility here. And we can look at in different populations from different studies, highly linked gene variants and where those gene variants actually fall in the structure of a gene. And again, this is all available programmatically as well. So how can these sorts of results be used? Well, one way in which these results are being used is by the construction of polygenic scores. A polygenic score is a single number that gives you an indication of the genetic predisposition for an outcome. It takes into account summary statistics from GWASs, of which I've shown you a lot now, and the genotyping of individuals in a data set. And so we may know that SNP number 1 is highly associated with whatever outcome. And so for people who have that SNP, they're more likely to have that outcome. And we look at an additive effect across the whole genome. Additionally, we can operationalize these by doing gene by environment studies where having 2 copies of a risk allele versus no copies might not mean anything in one environment, but means quite a bit in a different environment. Or it might be inversely related in 2 different environments. And so is there some sort of, at this point, I'd like to make a little call to action, tell you about the resources that we actually have so that you could use this in your own research. And so the first I'd like to draw your attention to is that we've posted all the genetic data for GWASs to a repository called dbGaP. This contains lots and lots of metadata about the study. It contains information about ethnicities and racial backgrounds, sampling structures, longitudinal designs, information about how the data was collected. And we have under our omics webpage, we have a slideshow on how to fill out the application to gain access to the information through dbGaP, and then how to download it for use with Sugen. Additionally, we also have polygenic scores generated for our participants for dozens, or rather, I guess, hundreds of different outcomes that you can download directly from AdHealth for such outcomes as cardiometabolic outcomes, anthropometrics, smoking behaviors, mental health disorders, self-rated health, math abilities, and so on and so on. You'll find all of this on our website under the code books for polygenic scores.